Imagine a bustling city filled with countless construction sites. At each site, workers labor tirelessly, following blueprints to assemble complex structures. Now picture this scene within every cell of your body. These microscopic construction sites are called ribosomes, and they are the protein factories of life. Just like construction workers rely on blueprints, ribosomes use genetic instructions to create proteins, the building blocks of all living organisms. Proteins are essential for virtually every aspect of life, from transporting oxygen in our blood to fighting off infections. They provide structure, regulate chemical reactions, and enable cells to communicate with each other. Without ribosomes, our cells wouldn't be able to produce these vital proteins, and life as we know it wouldn't exist. Ribosomes are like tiny machines, meticulously translating genetic information from DNA into functional proteins. They are found in all living cells, from the simplest bacteria to the most complex animals. Understanding how ribosomes work is crucial to unraveling the mysteries of life and developing new treatments for diseases. In the next section, we will delve into the fascinating structure of these molecular machines and explore how their intricate design enables them to carry out their essential task of protein synthesis. Ribosomes may be small, but they're far from simple. These intricate machines are composed of two main parts, known as subunits, which fit together like a puzzle. Each subunit is made up of a special type of RNA called ribosomal RNA, rRNA, and a variety of proteins. The smaller subunit acts like a decoding device, reading the genetic instructions carried by messenger RNA, mRNA. The larger subunit functions as the assembly line, linking together building blocks called amino acids according to the mRNA instructions. Interestingly, the size and composition of ribosomes differ slightly between prokaryotes, bacteria and archaea, and eukaryotes, plants, animals, fungi. Prokaryotes possess 70 S ribosomes, while eukaryotes have slightly larger 80 S ribosomes. The S refers to Svedberg units, a measure of sedimentation rate which reflects the size and shape of the ribosome. Despite these minor differences, the fundamental function of ribosomes remains the same across all life forms: to translate genetic information into proteins. This remarkable conservation highlights the essential role of ribosomes in the grand scheme of life. In the following section, we will explore the fascinating process of protein synthesis, where ribosomes take center stage as the master conductors of this intricate molecular symphony. Protein synthesis, also known as translation, is a fundamental process in all living cells. This intricate molecular dance involves several key players, with ribosomes acting as the central stage where the magic happens. The process can be broken down into three main steps: initiation, elongation, and termination. Initiation marks the beginning of protein synthesis. The small ribosomal subunit binds to the mRNA molecule at a specific start signal called the start codon. Next, a special type of RNA molecule called transfer RNA, tRNA, carrying the first amino acid, binds to the start codon. Finally, the large ribosomal subunit joins the complex, forming a complete ribosome ready to begin protein synthesis. Elongation is where the protein chain grows. The ribosome moves along the mRNA molecule codon by codon. Each codon codes for a specific amino acid. The tRNA molecule carrying the corresponding amino acid binds to the ribosome, and the amino acid is added to the growing protein chain. This process continues, adding one amino acid at a time until the ribosome reaches a stop codon on the mRNA. Termination signals the end of protein synthesis. When the ribosome encounters a stop codon, it signals the release of the completed protein chain from the ribosome. The two ribosomal subunits then separate, ready to begin the process again with a new mRNA molecule. Throughout this intricate process, the RNA within the ribosome plays a crucial catalytic role. It helps to correctly position the mRNA and tRNA molecules and facilitates the formation of peptide bonds between the amino acids, ultimately leading to the creation of a functional protein.